It looks like we're starting with an online question. Please, Lisa.
Our first question is for Dr. O'Connor. Has the ETT addressed botanical drug development difficulties? Thank you.
Yeah, thank you for the question. So no, we don't have any current projects related to the botanicals. Obviously, it's an active area within CDER and OPQ, but not through the emerging technology team at this time.
All right. Microphone number 2, stand up nice and close for me if you would, please.
Hi. My question is for Sarah. As you mentioned in your talk about multi-attribute method, LC spec systems are generally not in the QC space. Thus, what level of equipment qualification is necessary for these systems for supporting biosimilar development and manufacturing, and how frequent is requalification needed?
That's a good question that I don't necessarily know the answer to. But it would, and if you're using it for characterization, then it's going to be a little less important than for if you're doing it actually in the QC lab. And I think it's going to be similar to any other instrument.
So when you're doing multi-attribute method in support, if it ends up being like a release for biosimilars, then it's going to be more important to do that then?
Yeah.
All right, and again, if there's any other questions here in the room, come on up to the microphone, but let's take our next online question, please.
Okay, this question is for Sarah. Do you expect end users to use accurate mass instrumentation or nominal mass LC, MS/MS instruments acceptable for the MAM methods?
So that's going to depend on what your intended purpose is. Generally, what we've seen so far are high-resolution accurate mass instruments. And in order to be able to perform the new peak detection function, you're definitely going to need high-resolution accurate mass. You can't really make that, you can't find those new peaks without that. In specific cases, you might be okay without accurate mass in for the product quality attribute part of it, but it'll depend on specifically what you're looking at and what your product is.
All right, microphone number 1, please.
Yes, so this is also for Sarah. So you mentioned looking at conventional methods versus MAM methods. Have you guys taken a look at using 2D LC to perhaps bridge a conventional LC method that might not be appropriate for MS or MSMS, and using the second dimension to then flow it into and try to bridge them that way?
We haven't actually done that, but that's a really interesting approach that I think could be useful for looking into the method comparison if that's something you want to try for yourselves.
Okay, microphone number 2, please.
Hi, my question is also for Sarah, and it's related to the previous questions too. In case that you is towards the regulation to accept that if you use the MAM method for release, then you will be able to substitute the other method that are used as orthogonal method at the moment, like when you put on the table which is covered, is going to be accepted?
So right now, this is still going through the emerging technology team. So there aren't any set standards at this point. We would need to work with you on a one-on-one basis and kind of look into what your process is and what you're proposing specifically.
Thank you.
Mm-hmm.
Okay, let's take an online question, please.
Our next question is for Salvatore. When out-of-specification results occur in continuous manufacturing, how do you assure that non-compliant material is removed from the process? Thank you.
Thank you. Hello. Thank you for this question. So this relates to the understanding and calculation of the residence time and residence time distribution across the process. So we need to understand exactly how much time the plug of material spend across each single unit operation, such that when we detect non-conforming material real time that don't meet specification, we can diverge out of the process this material for a specific amount of time such that this is not carried forward. So it's relatively and importantly dependent on the residence time of your process.
Okay, microphone number 2, please.
Yes, I'm Bob Gaitis from Corning. So I think back to an article that Dr. Woodcock wrote in 2012 related to sterile injectables and the need to address drug shortages. And in that article, they talked about the difficulty in getting companies to adopt new technologies. And obviously the ETT and OPQ are critical to helping to move new technologies forward. Corning has participated with the ETT on in our Valor glass vials. The ETT has been very helpful in moving this product forward through dealing with in conversations with FDA reviewers, et cetera. And in fact, the research that the paper you presented on glass vials, vial C there was Valor glass in comparison to borosilicate vials. So my question gets, maybe for Mike or Larry, and I don't want to put them on the spot necessarily, but there was talk yesterday about the ETT 2.0. Obviously, ETT has done a lot of work around continuous manufacturing and 3D printing and helping to facilitate and promote those kind of technologies. Is there an effort or a thinking about how products that have gone through the ETT program, the ETP program, that they've graduated and that they actually help to improve quality and they may help to address drug shortages, and to get that information out more broadly to the public? And again, I'm not sure that's necessarily for this panel, but perhaps it is.
So I can just talk a little about I think we've talked a lot within ETT and ETT 2.0 about the concept of graduation. I think it's a little bit different than what you articulate, which is also very important. You know, in our head, what we mean by graduation is that technology is becoming sufficiently mature. See, we've gotten a lot of sponsors for doing a lot of applications that it no longer needs to go through the ETT. You know, we gained enough knowledge about that process. We've also draft guidance maybe, you know, and we can kind of transfer that back to the review divisions. Not just blindly, you know, there's training, there's taking all the learnings, all the discussions, making sure that knowledge transfer happens, documenting everything we've done as far as research, the conversations, the applications, and then moving that back to the normal review paradigm. So we are currently developing processes to help us do that. And that really helps us moving forward because, you know, what's new today is not new, you know, 5 years from now. And you know, the ETT is a small cross-functional team. We can't keep hanging on to the things that we did 5 years ago. We need to kind of keep moving on. And we have capable review divisions that are willing to take that on. So that's what we've been talking about as far as graduation ETT 2.0. So hopefully that answers maybe part of what you're talking about.
To get at Dr. Woodcock's goal of helping to incentivize the uptake of new technologies, I'm not suggesting that that FDA or OPQ or ETT should be promoting or endorsing particular products, but be able to communicate what they were able to assess during their review in a more public way. I think that could be helpful.
If I can quickly expand on this from the industry perspective, I can tell you that there is a strong interest from hospital, from non-profit organizations serving hospital, to use continuous manufacturing as a mean to produce this drug that is short supply, mostly sterile injectable. Continuous manufacturing is definitely not only a solution for the solid dosage form, but definitely as well for a sterile injectable drug. And this could be a viable solution for actually resolving this problem. Thanks.
And I think, Larry, did you have something to add to that?
Yeah, I mean, what we will do is, if appropriate, we will issue a guidance, and if we need to communicate broadly, you issue a guidance and those things to communicate.
Yeah, thanks. Jeff, I just wanted to add as well, this is Mike Kopcha. You know, as you know, these technologies become tools in a toolbox. You know, we evaluate them, we assess them, we do publish on them as well in peer-reviewed journal articles, things along those lines. It's up to the companies to do their business assessment and figure out what is the best technology or tool to use, depending upon what their needs are and what issues they're trying to address. Continuous manufacturing, as Salvatore had pointed out, is only one tool to maybe bring down drug prices. But again, companies have got to do their due diligence in terms of making the business case, selling it to their upper management, and then saying yes or no. But we leave it up to them. We can't promote technologies other than the work that we've done. Let's face it, we're a government agency. We're not in the business of marketing. So we leave it at that for the companies to make those decisions. So thanks.
All right, microphone number 1, please.
Warren from Camp Pacific. The question is for Dr. Mascia about continuous manufacturing. Actually, it sounds like very good technology, but I just want to ask, how do you do the OS investigation? You have so many different factors, and do you have an actual example that you have actually conducted out of a specification investigation? In the conventional or current guidance, there's phase 1, phase 2. Now, how would you do that? Thank you.
Yeah, so again, the reason how we identify out of specification material is that we looked at the obviously specification they require for the specific product for both API and drug product. Then when we develop the process, we perform a lots of analytical testing offline with conventional offline technique like HPLC, GC, X-ray, and then what we do, we match those data with the inline PAT sensor that we're using to monitor this quality attribute, such that when we integrate the system and we run the process, we can identify this out of specification material, so material which is not conforming to specification, through this inline measurement. I show you before example using the React IR for looking at the purity. We use the NIR for looking at residual solvent, particle size analysis through laser diffraction, for content uniformity, the NIR as well. So we have inline tool that allow to determine whether or not this material is conform to specification. But it's based on a priori development work to match this spectroscopic data with the analytical technique.
So basically, you have this inline and online technology to really, kind of doing this real-time quality control. That means like you probably actually don't have the OS situations then, because
well,
if it's out of
And for the sake of time, you guys are going to have a wonderful lunchtime discussion. But we've got a lot of people waiting with a lot of questions. Can we have another online question, please?
This question is for Dr. Mascia. How does a company assure that the recovered solvent that they feed back into the process does not have trace levels of impurity that will negatively impact the process?
Thanks. This is a very important question because we have seen actually issues with recovering solvent, bringing them impurity, actually altering final quality of the product. So we actually have measurements. I haven't showed this because it wasn't time, but we do measurement on the solvent. So we basically first separate. These are two solvents. We first separate them. Then we do some distillation actually to purify them, and we can show that we recover a high purity, high yield solvent from the system. So we do basically a refractometry measurement, an inline measurement of purity of this recovered solvent before we fill it back into the process. So it's basically inline analyzed before we actually reuse this. So we test this on a real time.
Okay. Microphone number 2, please.
Yes, my name is Shantini Jayaraja. I'm from BioAD Solutions. My question is for Sarah. I was wondering whether you can elaborate a little bit on the quantitation strategies for glycopeptides or charge variants. I understand for modified species in very low levels, yes, mass spec is a good instrument, whereas glycopeptides or charge variants, we have established methods for quantitation. How are the, first of all, the first question is it still going to be relative quantitation or will you be using standards? The second is about the performance of this quantitation, whether you have any data currently available.
So MAM is entirely relative. It is always comparing a modified peptide versus an unmodified. Or actually the total. So you'll have, for glycans, you'll look at all of the glycan species you're analyzing, and you'll look at a particular glycoform over the total glycan species for your quantitation, and it's relative. And the second part was the performance, I think. I mean, so through our method validation, it seemed it's generally pretty reproducible as far as that goes, and because you're using high resolution instrumentation, you're pretty accurate at what you're looking at.
Yeah, my thinking was that you also suggested that you will compare to traditional methods, conventional methods. So when you compare them, do they still outperform the conventional methods?
I mean, I think that's going to be on a case-by-case basis for whatever method you're looking at. Again, you're looking at different things, so it's not a one-to-one comparison. And performance-wise, I think they're generally fairly comparable or better depending on what you're looking at. It's not worse.
Sounds like another excellent lunchtime discussion. Another online question, if I could, please.
Okay, this is for the panel. Have there been any discussions about applying a second generation of this approach to biologics?
Hello, I can start here. I think that there is a lots of similarity between, I'm talking about the continuous manufacturing process application from the small molecule and the biologic. Obviously, we're talking about different requirements, but the philosophy and the control strategy, the way you think about this, are very similar. Obviously, for biologic, you have a perfusion reactor already continuous and implemented, and the challenge is more downstream. And I think that some of the learning and the new technology developing on the downstream can also find application for the biologics. Obviously, it's not yet there as the progress that we made with the small molecule on an end-to-end integration, but I think that the concept of thinking about this and applying this are actually transferable between the two.
Yeah, I would also say, we've had through the ETT conversations with different sponsors looking to implement continuous manufacturing or elements of it for biomanufacturing, and we have active research projects in the area, both internally and externally.
Anything to add?
Oh, I don't think this one really applies to me because I'm already doing biologics.
All right, there we go. And that takes us right up to the end of the time we have for the panel. Please help me thank our panel for sharing their ideas.